Dr. Ben Newman, coronavirus researcher. No, not you, Darwin. Yeah. uh, this is Ask Dr. Ben, where I try to answer your questions. And how do you like my new ring lights? Much brighter in here. Yeah. I forgot on that last video, I left all this stuff up at the side because I meant for this one to be the next one, and I just forgot. So anyway, here we go. The next one is from Barb, and welcome back. Yeah. Let's see. Hi, Dr. Ben. Israel just had their first case of flurona. Yeah, and so did California. Yeah. A pregnant woman has both the flu and COVID. Can these two viruses merge or become some one other monstrous virus? That's a decent question. Yeah, thank you. All right. So, first of all, let's look at this thing. So, yeah, I don't know. We're living in a Cheech and Chong sketch or something. Flurona is what we're calling it. Fine. It is having influenza and SARS-CoV-2 at the same time. And so what does that mean? Like, can this happen? Sure. SARS-CoV-2 infects things that are called type 2 pneumocytes, which are the ones that make a soapy substance at the bottom of your lungs that helps the air slip in and out a little bit better. And flu, I think, hits the ciliated epithelia, which are these cells that have little, kind of like little tiny tentacles on the outside. And they're much more common in the lung. But I think there's a particular subpopulation of those that different kinds of flu prefer and whatever. So, yeah, the different cell types. Being infected one would not necessarily mean you couldn't be infected with another for any reason that I can think of anyway. What does that mean? Well, if you get infected with one, you'll make a macrophage response. You'll start to have these immune cells that are embedded in your lung wake up and start to just go around and randomly fight. And the thing with macrophages is they're dumb, but they are, kind of big and kind of tough. And so they can do a lot of damage to any infected cell. And so if you have one infection, you may wake up enough macrophages, at least in an area, that you're going to be kind of protected from another infection, or it won't maybe get out of hand. But at the same time, there's no reason why those two, viruses don't end up in different parts of the lung, in which case you just have two infections starting around the same time, and it's totally possible. In the past, we have good evidence of people with two different coronaviruses at the same time, or with a coronavirus and influenza virus at the same time, or coronavirus and pneumovirus at the same time, or metapneumovirus, or whatever. It can be done, yeah. What are the outcomes? Generally, we only have small numbers of people that have both of these things at the same time. All you can say is it's not going to be any better for the patient. It's probably going to be worse, but in some kind of non-specific, vague way. So, yeah, flurona. I don't know. Anyway, now, part 2, flurona. Yeah, really? All right, maybe. Here we go. So, you can check how the U.S. is doing on flu cases. We have a tracker, and there are similar trackers you can find in the UK and in Europe. Chinese CDC may even have something like this. I'm not sure. So what we've got here are flu cases, and these are actually positives that have been tested in a clinical laboratory. Now, that's a key point in understanding any one of these numbers. And the numbers you can see here are, we're doing about, around 4,000 per week right now, as of this past week. So, yeah, that's actually reasonable for the start of a flu season. It looks like flu season may be back. Hooray. Yeah. But those numbers come in a very specific way, and it's actually the same way that we get the COVID numbers. So if you take something like a rapid test and you test positive and you tell somebody about it, that's okay, but that does not go into the official statistics, because at least in the US right now, the official statistics have to be done with a specific set of tests by one of these laboratories. So these are called CLIA laboratories, and there's a whole bit of legislation, and there are a lot of really competent, really high-end laboratories in the world that are not allowed to be CLIA certified. Essentially, it doesn't say so in the rules, but in the application of the rules, you effectively have to have a medical doctor on staff in order to have a chance to become CLIA approved. And if you're mostly doing animal work, even if you're doing the same sorts of tests, you can't be CLIA approved. This is what people have found out. So you doing a test on your own is fine. You gain knowledge, but the world will never have that. That little statistical marker will not go up by 1 because you didn't do it in a CLIA lab. And so you get places where they'll do the test and then they'll send it to a CLIA lab if it is positive, and then they can get confirmation of the positive. That's like a workaround, but you basically have to run the same sample twice, and it's kind of a waste, but it's for bureaucracy. So yeah, go for it. And there are or were reasons behind this, but I don't think the reasons for those particular interpretation of the regulations have a whole lot of merit. But again, yeah, I'm not a politician or a lawyer, so whatever. Yeah. All right, so that's flu this year, and so yeah, 4,000 cases a week. Pretty good. What's a typical year look like? So let's go in the Wayback Machine, the 1819 flu season, which is the last one that's untainted by COVID. And so here we can see that when we get to the peak, we would be expecting something like, I don't know, 14,000 a week. That would be about right. And there will be a lot more cases than this, but 14,000 of them will have been done with all the proper paperwork, all the T's crossed and I's dotted in such a way that they are allowed to be reported. And so it's actually the same with COVID cases. So here we've got, current 4,000 a week. COVID cases, if we look for COVID cases in the whole U.S., which is what I have it set on here, all regions, you can see we're doing, there we go, 7-day average is 584,000 per day, which means that you have to multiply that by 7, and so you get some enormous number. Yeah. What is that? About, neighborhood of 4 million cases. So there's 4 million COVID cases right now per week versus 4,000 influenza cases per week. Occasionally, the two will meet just because corona is everywhere right now. And it's not particularly surprising. There's no way these can recombine. They don't have any parts that are similar enough. Recombination only really works when you have two things that are more or less the same, and preferably for long stretches. And then you can get, as the virus is copying one, it'll run into some kind of problem, switch to the other, and maybe even switch back again. Now, my entire PhD was spent futilely trying to get coronaviruses, which famously recombine all the time, trying to make a coronavirus recombine with something that it didn't like. And we put thousands of bases before it and hundreds of bases after it. And we just, you could sit there and watch. And every day I'd start it up and there'd be a couple cells. We were trying to put that green fluorescent protein in. And then the next day there would be maybe a tenth that number. And by the next day, maybe one cell is left. And then by the next day, it's gone. The coronavirus, we gave it all these different chances. We gave it all kinds of incentives. We put in antiviral resistance markers to try and just get it to recombine that in, just so we could get to recombine with something. Wouldn't do it. Yeah, did not like it, wouldn't do it. Coronaviruses recombine very easily with other coronaviruses and with nothing else that I have ever encountered. Theoretically, it could happen, but the other thing here is that flu viruses work kind of backwards to the way coronaviruses work, in that they just start off with like the opposite strand. You know how there are two strands of DNA that run in opposite directions, or RNA? Flu start with the bottom strand, corona start with the top strand. And I don't know, the proteins that make them are really pretty different. The substance of these are really different. There's no reason why coronavirus would recombine with flu. It would be more likely to recombine with one of the thousands of other messenger RNAs that are around it in the cell all the time when it's copying itself. But even that doesn't ever happen. So yeah, really they just recombine with other coronaviruses. because you probably got two coronaviruses that are sitting there being copied at the same time, and it's easy to just sort of swap over when you've got basically the same. You just it's like passing a book between two people. So no, I'm not worried about flurona yet. Yeah, I don't know. Famous last words, right? I don't think there's even all that much flu right now to need to worry about it. But at the same time, last year we had no flu season because people were masking and distancing, and this year we ripped the mask off and done all our holidays in person, and no kidding. Coronavirus numbers are higher than they have ever been, ever. And we have a tiny bit of flu, so there you go. Yeah. Corona's still the real problem that needs to be knocked out. And we don't have enough information to say whether having flu and coronavirus is different in any specific or meaningful way, or whether it's slightly worse. It's not going to be better, but it could be slightly worse. But there's no you need to have several hundred or several thousand people with both, and you could get all the people together in the country and not find that, I think, at the moment. So it may be some time, maybe some years before we have a decent answer to that. But for now, I wouldn't worry about it. I don't think it's a big thing. I do think Corona is a big thing. And yeah, that's what I'm worried about. All right. Thanks very much. This has been Ask Dr. Ben.